Hi, it's Paul Sunga, Langara College. This is a truncated version of a talk I provided for my pathophysiology students. It's a bit long and complicated, so I've shortened it down just for this purpose, for easier public access to this information. COVID-19, summary of emerging findings as of March 23rd now, 2020. Just an outline of the talk. I'm going to talk about the virology of SARS-CoV-2, especially not so much about the natural history, which is well published, its origins, but a little bit about the phylogeny, the current phylogenetics, as well as structural and virulence factors, which are relevant for everyone. Practical details of the chain of infection, which means how the virus is transmitted, its portals, and the risk factors involved. Thirdly, we'll look at the pathogenesis, especially a few things, the key features of the tissues involved, as well as diagnostic highlights and the overall clinical features. And then interventions, questions, and observations about the current epidemiological situation, and a few comments about the prospects for the near future. So the virology of SARS-CoV-2 is that this is, of course, a coronavirus. Most people know that. It initially presented as a cluster of cases of pneumonia due to an unidentified virus. So viral pneumonia is something that does happen. It's not usually the most serious form of pneumonia. This happened in Wuhan, China, and it was traced back to the open-air market. People had attended this market in late 2019. So subsequently, the virus was identified as by using viral genomic analysis that was done by Chinese researchers. It was found to be similar to a virus in other mammals, especially that carried in bats. Now, a number of viruses are carried by bats. It's a reservoir for a wide variety of viruses, actually, not only coronaviruses. However, we also have some information that indicated that a reservoir of this virus is found especially in pangolins, even though an initial paper found that it might be similar to snakes. This was the initial report, but it's not as likely as something like a pangolin, which is a closer relative to humans and bats. Coronaviruses are found in a lot of different animals, including birds, camels, bats, civets, mice, dogs, cats, and others as well. So the phylogenetic analysis, I'm going to show two things. One is analysis which has been done on other SARS viruses that indicate their class. That you can actually place viruses into the same type of Linnaean taxonomy as living organisms. Well, probably important to take a pause here so that people realize that viruses fall into a gray area between living cells and inanimate material. They are very simple, obligate parasites. They can't reproduce or metabolize on their own, so they require a host cell to be able to hijack, and this is what they do. However, based on genetic analysis, you can still organize a taxonomy. In the case of coronaviruses, they're in the order Nidovirales, in the family, which is called Coronaviridae, and the genus beta coronaviruses. Then they're divided into serological groups. According to the serological groups means that according to the antibodies that the antigenic differences that are identified using antibodies, beta coronaviruses fall into 4 groups, actually. So this paper by Gorbalenia, published in 2020 in Nature Microbiology, shows some of the key genetic aspects. The first diagram on the top just shows areas that are identified as areas that can be sequences that can be used to identify the virus, and that's the basis of the diagnostic procedures that are used with PCR. Currently, I'll get into that later, but you can see the phylogenetics on the next diagram down, which shows SARS-CoV and SARS-CoV-2 fairly closely related with other viruses. And then in the bottom, you can see a number of viruses that are linked together into these different groups. And you can also look at the different names of the viruses that are found in different animals, including bats and mice, rats, etc. But possibly even of greater interest, looking at this work that's been done on the genome of the virus, is current work that's ongoing. I hope I can go to this. Coming to something more concrete, though, looking at the viral structure, this right-hand picture shows an electron micrograph, color. They're in black and white, but colored just for contrast. And you can see the characteristic surface spikes that are found on coronaviruses, hence their name, corona, crown. These surface spikes are what they look like, the spikes which attach to the host cell. In this case, it's respiratory epithelium. We know what the receptor is for these, and we also identify researchers have identified the spike protein S it's called, as well as a number of other proteins that are found in the capsid. So this is a typical, for those who are unfamiliar with virology, it's a typical viral particle. It's made up of a quite a simple structure compared to cells. Even bacterial cells are much more complicated in terms of their structure, especially their outer structure. But viruses have an outer capsule which is made of proteins. There's just a handful of proteins. The genome is a single strand of RNA. Now, most people have heard of DNA, but this is RNA, which is a closely related compound. It's probably the original genetic material in terms of living things on Earth. It's less stable. It's found to be the genetic material in a whole class of viruses that we call RNA viruses. So this virus is a large enveloped single-stranded RNA virus. There's a few that have double strands, but this is a single-stranded. It has 29,811 nucleotides, which are the units that make up the RNA, and it codes for 9,860 amino acids, which are the components of the proteins that form the virus. So these proteins that the virus has on it are the proteins that are needed for viral replication, for binding. We call them virulence factors. These virulence factors are the things that undergo changes as the virus mutates and give it its variations in its ability to bind and ability to replicate. So when the virus binds to the host cell, the human tissue, it binds to a particular protein called the ACE2 protein. So this is a protein that's found in the cells of the mucosa and the respiratory tract, especially in the cells of the lung, and also in the myocardial tissue, the heart muscle. ACE stands for angiotensin converting enzyme. It's an enzyme that is involved in regulating blood pressure. Now, there's actually two versions, angiotensin converting enzyme protein 2 and angiotensin converting enzyme. The one that is highly active in people and that is regulated by drugs that are given to hypertensives called ACE inhibitors is the other ACE. But there is a link here between these two. There is a thought that people who are taking these ACE inhibitor drugs are more susceptible to infection, and more susceptible, I should say, to complications as well. So then looking at the docking protein on the virus and the receptor for that docking, knowing these things, of course, helps to give clues as to what type of medical interventions, in other words, drug interventions, might be possible. Also knowing the surface proteins, of course, is what is needed to develop vaccines. For the uninitiated, this rather complicated looking picture from Sunga et al. They've been investigating coronaviruses for some time. This is a typical picture of how viruses actually cause damage to cells. What viruses do is, if you start at the left upper area here, is they bind to the receptor. They're internalized into the cell. Once they're internalized, they essentially cause copies of their own genetic material to be made, which then gets used to make viral proteins. It's called the process of transcription. Once these proteins are made, you have the elements of new virion particles, and these particles have to go through some modification. They use the host cell to do that, and once they've done that, they are released from the cell. They bud out of the cell. So damage to the host cell, this whole area is inside the host cell, so this might be your respiratory epithelial cells, which are the cells that line your respiratory tract. All the damage that happens occurs because this process is happening abundantly inside your cells. And once that happens, the membrane is disrupted as budding off occurs. You can imagine that the normal processes of the cells, metabolism, its housekeeping if you like, its chemical processes, are going to be disrupted by it being taken over by this virus particle. You see on the right side a closely related virus. It's called the MERS virus, the Middle Eastern version of this beta coronavirus, which is highly lethal. That's been found at a reservoir in camels. But anyways, you can see it's a very similar type of pattern of replication, of internalization, hijacking, and budding. Now, all viruses essentially work this way, but there is differences in the details of how they do this. And in this case, we have a pretty good idea how this occurs. This also is going to provide clues as to how drugs, medications can interrupt the cycle and stop the process. And this is, of course, many of the antivirals that have been developed, quite a few of them developed after the HIV epidemic, and are specific for HIV, which is also an RNA virus, by the way, are drugs that interrupt various stages of this cycle. And as you may know, highly active antiretroviral drugs have changed the course of the HIV epidemic, at least in terms of death. So a practical issue, looking at the chain of infection of COVID-19, what is meant by a portal of entry is, of course, the way in which the infectious particle, in this case the virus, gets into the body, gets into your system by breathing it in through the nose or through the mouth and into the funnel at the back of your mouth, which is the oropharynx. The tissue that lines that is called the pharyngeal mucosa. So once this will give a sore throat, an irritated throat, as the virus binds there, and as it does so, it's also starting to replicate, and as it does so, it's also moving, being passed by fluids into the lower areas of the respiratory tract, in other words, the lungs. So this is presumed to be the main mechanism. In some cases, when you look at completely other viruses, they can enter into tissue and be passed along to other areas of the body through blood or through nerves. The actual passageway from the pharynx and down into the lungs is assumed to be that people either breathe it in or that they have the virus passed by fluids into the respiratory tract. Anyways, viral DNA in terms of hardcore findings, where people have looked for, have sampled different tissues. You see the list here. It includes in the lungs. These are hospitalized patients now. This is work of Kai, Sun, et al. You can see the reference there. it was published in Emerging Infectious Diseases very recently. So in this sample of patients, they found 14 to 15 have RNA virus, which means the viruses are present in 93% of the cases. In sputum, 72%. In the nose, in lungs on biopsy, in other words, you take a sample of the tissue, 46%. Throat swabs, but also feces and blood, feces 29%. So this means that the virus actually seems to get into the GI tract, the gastrointestinal tract, and then in some cases it's going to proliferate into the blood, which is called viremia, and pass to other tissues, presumably. So the portal of exit is primarily respiratory. The fecal route is probably, but fecal transmission, as I said, is also possible. Transmission via fomites is assumed. At this point, probably more information is needed to support that. And there's also little evidence of vertical transmission. Vertical transmission is passage from the mother to her offspring. So what are the risk factors? Risk factors means that if you studied at the population level, you'd find that people that have certain features or certain habits, or they have physical features or medical features, that all of these things, or each of them, of course, when you're analyzing it, increases the probability of getting an infection. So risk factors for COVID-19 include advanced age. Most of the people who are being seriously affected in Italy, for example, are over the age of 80 by far, especially for hospitalization and especially for succumbing to the infection. Exposure, close contact with somebody who's infected. Smoking, by presumably decreasing the capability of the respiratory tract to defend itself. We know that smoking alters the defenses. Comorbidities. Comorbidities are underlying conditions, like chronic conditions that alter the ability of the system to fight off a viral infection like this. There are different mechanisms at work here for different comorbidities, but we know, and I put these in roughly the order of how important they are based on a number of papers, including the paper by Zhu, which is mentioned here. Hypertension, HT is hypertension, high blood pressure. Diabetes, mostly we are talking about diabetes mellitus type 2, coronary heart disease, atherosclerosis, and ischemic heart disease, all those are components of that. COPD, which means chronic obstructive pulmonary disease, such as emphysema and chronic bronchitis, are the principal things that are involved there. People who have COPD are, by and large, in Canada anyways, mostly as a result of smoking. It's classic smokers, lung and cough. Malignancies, ongoing cancers for which people are being treated, chronic kidney disease. The idea of carriers is that people carry the virus and are able to transmit it to others. And the two types of categories that you can consider here are asymptomatic carriers and symptomatic carriers. In other words, people that don't show any signs, aren't sick themselves, are asymptomatic carriers. So there's two types of asymptomatic carriers. Those people who are infected but haven't yet started manifesting. I mentioned the 5 days period during which people might be infectious, but they might not know that they're going to be sick. And the second one is convalescent carriers, which are people who did have the infection. Now they may have very low level subclinical infection. They're getting over it. Their defenses are defeating the virus, but they're still able to transmit it to others. Symptomatic carriers are people that actually, of course, manifest it. They have symptoms and signs, but they still feel okay. and they're still infectious, and they pass it to other people. The thing that's caught the public's attention, of course, is that younger people seem to be able to fight this much better just based on the data so far. But of course, they can pass it on to people who are more vulnerable, such as older people. The last thing to mention here is the thing that, again, has caught the public's attention, which is the idea of super spreaders. Some records indicate, there's anecdotal evidence really, of individuals who are able to infect many more people than simply the average of about 2.2. There's a case of an individual who came back to Hong Kong from China, who from Wuhan area, from Hebei, who moved into his apartment, and then almost everyone in the apartment was affected after that somehow, even without being in close contact. And the second notable case is the young man who returned to Lombardia in Italy, who went to healthcare clinics because he didn't feel well. He wanted to be diagnosed for COVID-19. Wasn't successfully diagnosed a couple of times, and seems to have spread it very widely among people in the healthcare settings and also in the public. Why are people super spreaders? There's a couple of different things that are possible to think about here as underlying mechanisms. One, is it possible that they have such a high viral load that it becomes aerosolized and that people now get exposed at a much greater distance? What higher viral load means is the number of particles per unit volume. In terms of the numbers that have been found, they found that people who are seriously ill can have up to 10 to 11 per microliter of virus, which is a very high number indeed. Some issues about diagnosis and the pathology. Diagnosis first. Diagnosis is done by taking a throat or nasal swab. It can be, of course, samples can be taken from other tissues, as I mentioned in the list, but even from blood or feces. But for most people who are screened, throat or nasal swab. What happens with this sample is it is processed in a process called real-time PCR, which is polymerase chain reaction. And what that is a way of amplifying the RNA, turning it into DNA, amplifying it, and using it for the sequence analysis, the type of thing that I showed with the data earlier with the phylogenetic analysis. They are essentially looking for a fingerprint or certain sequences that are unique to the virus that allow it to be identified. It's a relatively fast procedure, but it is a technical, highly technological procedure in that it's molecular biology technique, really. And so it has slowed the ability of the medical system to actually screen on a wide basis. The more common way to identify all kinds of things, including infectious diseases, bacteria, and viruses, is using serological analysis. The problem is that we don't have test kits available right now. Serological analysis means that you look for the presence of antibodies or antigens in the person's blood. You take a simple test. It's quick. There's even rapid tests that can be used of saliva, for example, for HIV, that can be done at home for just a little strip of paper that shows a color. Not there yet. This would, once these serological tests are available, they will expand the testing capacity greatly and also make it much less expensive to do testing. ELISA is simply the enzyme-linked immunosorbent assay. It's just a matter of being able to visualize these positive tests. Pathogenesis. Pathogenesis means the process or the pathway of the disease, of the infection, what happens to it in your body. A few things that are notable. One is that among a large portion of people who are hospitalized, they are found to have damage to their heart. Acute myocardial injury means damage to the heart. Essentially, the test you use is looking for heart proteins in the blood, which means that heart cells are being damaged and leaking their stuff into the bloodstream. This has been quite a common injury. Again, it's probably because of the presence of the receptor in high abundance in heart. Second point to make here is that common to a number of other serious viral infections, including H1N1 flu, elevated inflammatory mediators, things that cause inflammation in your body. There's indications of rampant inflammation that happens in the lungs, which is the basis for the flooding of the lungs that happens. It's called an inflammatory storm or cytokine storm. These are just the chemicals that are involved in that. Neutrophils are cells that are a signal that inflammation is going on. In terms of the lung itself, the histology, the examination of the tissue, there's presence of abundant fluid, edema, as well as what are called proteinaceous exudates, is again indication of damage as well as inflammation going on in the lungs. So the blood vessels become congested with fluid. There's also, the lungs will attempt to try to repair. They become temporarily scarred as fibrinoid material that are found. Cells are evidently being damaged, of course, by the viral replication, and it gives rise to these abnormal cells, multinucleated giant cells, as these cells fuse together. And hyperplasia, which is a compensatory, what happens is the cells, as they're injured and damaged, they start to proliferate to try to fill in the gap for the cells that are lost. And this is from a paper of Chen, but it's also a number of other papers that I've put in there. It's not solely that paper. This shows a chest x-ray in which you can use to observe areas of fluid involvement and scarring that's going on in the circled areas. If you're not used to looking at x-rays, it seems like a kind of a subtle change, but it can be a lot more clearly, I should say, with CT scan. And CT scans give a lot more detail, and the presence of opaque areas that are present in these CTs. Essentially, these pictures are looking down at the lungs, and the areas of injury appear as, opaque areas that indicate that there's liquid inflammation and damage occurring in the lungs. Ground glass opacities is the term that is used. It's one of the hallmarks of this type of infection in hospitalized. This gets more into the type of things that are of concern for hospitalized patients. So this left-hand list, dry cough and runny nose, rhinorrhea, fever, myalgia, which is muscle aches and malaise, feelings of feeling lousy, feeling sick, basically is what that means, headache, dyspnea, tough breathing, so having a hard time getting breaths. And again, if there was a CT scan, the presence of ground glass patch opacity is present almost always, especially if there's the tough breathing to dyspnea, which is going on. Complications found in hospitalized patients, almost all of them are going to suffer from interstitial pneumonia, the flooding of the lungs, which happens with infections. This is fluid in between the blood and the air spaces, which makes the passage of oxygen and carbon dioxide, which is exchanged in the lungs, less efficient. And so the patient starts to suffer from not having enough oxygen in their blood, which is called hypoxemia, and subsequently in their tissues, they become less oxygenated as well, which your tissues need oxygen, of course, in order to make energy to keep you alive, to do the jobs that they have to do. But anyways, interstitial pneumonia, ARDS, which is acute respiratory distress syndrome, it's a particularly dire lung disorder. It has a very high mortality associated with it. Traditionally, something like 40% of people, up to 40% of people will die if they have ARDS. Their lungs become so dysfunctional that they essentially suffocate with flooded lungs. But 29% of patients have ARDS who are admitted patients. Acute myocardial injury, as I said, heart involvement in 12%. Acute kidney injury, sepsis, which means that there is other types of infections which take the opportunity to move in once the person is in this compromised situation. And septic shock, which is the result of sepsis, in which the presence of overwhelming infection causes the person to go into shock. And again, very high rate of mortality associated with sepsis and septic shock. And before they actually succumb, their organs will fail. It's called multiple organ dysfunction syndrome, MODS. And case mortality rate overall, 2.3%, but I'm going to talk about this a little bit more because there are differences. There's quite a variation in the case mortality rate. The number of people who die per person infected is quite different in different jurisdictions presently. So the complication is the way people will get into trouble, and it's often people with, remember, those comorbidities that if they have compromised defenses, compromised lung function, cardiovascular morbidities such as hypertension, congestive heart failure, ischemic heart disease, all of those people are going to be much more susceptible to getting into trouble. And among those hospitalized patients, we're talking about usually people who are older patients that end up in this situation on the right side. Different categories. Public health interventions, of course, mean the things that are going on now, which is social distancing and shutting down public places where people can transmit more easily, all of these measures. The only thing I wanted to say about this is the observation that public health, of course, is government writ large. It can only be led by governments, really, for the public, for the broad public. And therefore, the role of government in the public sphere is something that really becomes important. It becomes very political in jurisdictions where people may not accept a large role for government, which means they may not like to be told what to do and what limitations they have on their personal lives. Of course, public health may not work as well. And in areas where people are used to governments, where they trust institutions and trust their government to do the right thing, it might be a lot more successful. So these are really cultural issues about governance, and it's a very interesting area, but too large a topic for this talk at this moment. Second issue is looking at prevention is the consideration of where vaccine research is presently. Some notable things are that coronaviruses in general do not generate strong immunity. Think of the common cold. You get colds year after year. Many people get them routinely. You can have several colds in the same season even. The scientific kind of view of this findings are that for coronaviruses like common cold, there's 4 viruses that cause common cold. Your immune defenses, as measured by your antibody, the presence of antibodies in your blood, is weak, and it disappears after a number of weeks or a month or 2. Therefore, trying to generate vaccines which give you memory and a strong antibody response is a challenge at the moment. There have been no vaccines that have been approved for SARS-CoV or MERS-CoV. That's not for lack of effort. There's been a number of research projects that have been trying to develop vaccines, and they haven't been successful so far. So the generation of a vaccine for SARS-CoV-2 is going to be Not a simple process. There is a candidate vaccine, however. It's a new technology. It's using RNA to vaccinate. No RNA vaccines have been approved yet for anything, so it's very new technology. Whether or not it actually is effective, it's in clinical trials right now in Washington State. Clinical trials means that you're doing a study of vaccinating some people and giving other people placebo and comparing the two to see whether or not the vaccine works. And it takes some time to do that, and results should be forthcoming, hopefully sooner than later. Then there's the issue of rolling that out. It's going to take some time, at least a year, if it's successful. So there's a number of questions there. Vaccines are not going to be a simple fix. On the therapeutic side, there are a couple of things to say here. One is about ventilation. Ventilators, there's been a lot of press about ventilators. Ventilation is essentially, just to give the background there, it's a method of sticking a air pipe down into the lungs to force air in, force oxygenated air in, to combat the lack of gaseous exchange that happens in people with these flooded lungs. And ventilation of this sort requires that the person is sedated and paralyzed so that their own breathing rate, their own urge to breathe doesn't fight against the ventilating machine that is pumping gas through their lungs. So there have been a number of improvements that have improved the survival rate with people using ventilators over the last 15 years. Suffice that to say that at this point for this talk. Antiviral medications, there's a number of them that are candidates. One of the premier ones is remdesivir, which has been used in combination with the old anti-malarial chloroquine. Results of those clinical trials are still coming. Initial studies on that have not been that promising, but hopefully some tweaking of the protocols might improve. And so a few other observations and questions about the epidemiological situation. The first one about China. The bright light here is that the epidemic seems to be arrested in China. There's very few cases that are emerging every day now, a handful of cases really, especially given the large population and the challenges with very complicated distribution of this large population among cities and rural areas. How widespread and how good their sampling is for testing is a question, but they have been able to bring a lot of resources and a lot of personnel to this challenge, and there seems to be, we hope that there's good success there, of course. Good sign is that the temporary hospitals in High Bay are being dismantled, or by now are probably already dismantled. In Iran, another so-called epicenter of the epidemic, problematic government response here, partly for good reasons because they've been under U.S. sanctions for this last period. It stressed the healthcare system. And secondly, the leadership of Iran has been paranoid about, suspicious about what the origins of the epidemic are. They've suspected a U.S. plot. But the long and the short of it is, data is uncertain. We have anecdotal evidence and lots of reports from physicians in areas, especially outside of Tehran, which indicate that there isn't really good data on number of infected, nor of people who are dying. There's a lot of people dying in Iran of COVID-19 that I think are not being reported. So it's a difficult situation, and it will go on for a while. The other notable issue is a couple of countries. Top of the list is Italy, which has had very rapid transmission and also a high case mortality rate. If you calculate just based on current data, or as of yesterday, This data shows about 4,825 deaths, over 53,578 cases. It's already way out of date in terms of the actual numbers, but the proportion today is very similar, about 9% for the case mortality rate, much higher than what was found in Wuhan with its 4% case mortality rate on the same day. So what is the reasons for that? Possibly there's an underrepresentation of the actual number of cases. If the epidemic becomes very widespread, of course, it becomes very difficult to keep up with testing with everyone, especially given the diagnostic test that's being used right now. Overwhelmed critical care capacity simply means that there aren't enough critical care beds to deal with all the needy patients, and so some of them are falling off the edge of the healthcare system. Anecdotally, again, from reports from physicians in Iran, this seems to be the case. A lot of them are completely swamped with cases, with critical care cases. Sorry, in did I say Iran? I meant in Italy. There are these reports coming out. The third point is the demography of Italy is that it has one of the highest concentrations of elderly people over 80 years old. I think globally it's the highest, along with Japan. Something like 80% of those who are over 80 require hospital care if they're infected. So that figure is easy to remember. It seems to be the ongoing case in Italy and also in Spain. The method of assigning cause of death in Italy, from reports of people who are signing death certificates there, they're very liberal in assigning the cause of death to COVID-19, which may not be the case in other jurisdictions in other countries, so it's sometimes hard to compare. In other words, if someone dies, did they die from heart disease? You know, they're infected, but they died of heart disease. Did they die of heart disease, or did they die of COVID-19? It depends on how you look at it, and how it's reported is going to rely on someone's judgment. But the other hypotheses, even more interesting, high smoking rates, high levels of air pollution, of course, alter the ability to defend against viral infections and bacterial infections in the respiratory tract. And the last one is what I mentioned when I showed you the genetic analysis that's presently being done. It shows that there are different strains in different places, and what we know about that is that there will be natural selection for high transmissibility and possibly high virulence strains in different places, and perhaps that's what's happened in Europe at the moment. So with that in mind, last thoughts. This is an efficient virus in terms of its ability to transmit. It has a relatively low mortality rate, means that most people are going to be infected and not die, and they're just going to transmit, which this is what I mean by an efficient virus. It means that in terms of its ecology, it's going to spread efficiently, and it's going to also mutate as there's more cases and more virus particles that can undergo mutation. Especially with RNA viruses, RNA is able to mutate more easily than DNA viruses. RNA is an inherently less stable type of macromolecule. So is seasonal variation a feature? It's hard to say. There are some people who are suggesting that it is. There's some data that came out of China that suggests that warm and humid temperatures may inhibit the spread of the virus. Yet to be seen. We know that summer colds occur. There's another coronavirus that doesn't seem to respect temperatures that well. And you would also expect the immunity of the population to be higher during the summer when people are outdoors more, that their defenses are stronger, generally speaking, especially in temperate climates like in Canada. Generally speaking, the immunity, as I said, is weak for coronaviruses, so that's another mitigating factor. And then the possibility exists that there will be several, even continual waves of infection unless herd immunity develops somehow. Herd immunity is the immunity of the whole population. So Herd immunity will occur either because of vaccination, mass vaccinations, as it does with many other things that are vaccinable diseases, or that people develop natural immunity as they are exposed. So that's my update. It's 45 minutes long, a little bit longer than I thought it might be, but good luck with this, and I hope this information helps.